Well, good afternoon, everyone. Thank you for joining us this afternoon for Race Oncology's special investor briefing. My name is Jane Lowe, and I will be your host for today's session. Joining me are Dr. Daniel Tillett, who's Race's CEO and Managing Director. Good afternoon, Daniel.
Good afternoon, Jane.
Afternoon. And also Dr. Rodney Cusack, who is Principal Scientist with Race. Hello to you, Rodney.
Afternoon, Jane.
Thank you for joining us. So Daniel and Rodney will step us through the presentation, which was released yesterday to ASX, where we saw a lot of the recent work around the mechanism of action for RC220 and IP coming together in an expanded and very exciting clinical program. So after we've run through the presentation, we'll open up the floor to Q&A. If you'd like to ask a question, you can do that using the Q&A function in the ribbon at the bottom of your screen. Just type your question in there, and we'll get through as many as we can in the time that we have. But for now, I'll ask Daniel to start screen sharing and to get us on the road. Thanks, Daniel.
Thanks, Jane. Welcome, everyone. This is quite a long and complicated talk, but hopefully it will be a very valuable and insightful talk for everyone. Really
Daniel, sorry, I'll just mention we've got your presenter view.
Oh, okay.
Yeah.
I'll stop. You
Maybe just a slideshow.
slideshow.
Yep. That's it. Well done.
Okay. Hopefully that's better for everyone. Yes, I'd say a long but hopefully very productive presentation. Hopefully you'll get a better understanding about why we've gone about making these strategic updates to our clinical program. Through the work that's been done by the team at Race, we've really made a lot of discoveries over the last 2 years, really coming to a head in the last few months around mechanism of action and the IP protection. Mechanism of action in particular has really opened up a huge amount of opportunities, and we've systematically gone through the process to identify what are the best opportunities for our shareholders, what can we do that will generate the maximum value, and that's compatible with the resources we have. So I'm not going to spend any time on the notice of disclosure. And really, this is the culmination of the continued evolution of Race Oncology. Race Oncology started in 2016, building on the discoveries and clinical trials run by Ledley Laboratories, went through an initial phase focused on AML alone, and then since 2020 is really focused on understanding bisantrene, understanding how it works, generating IP around the formulation, solving the problems of the past, and making sure that we actually have something valuable at the end of all this sort of activity. And we've been incredibly successful, far more successful than I could possibly have imagined when I joined back in 2019. But everything that we've worked on has been a success to the point where we are today, looking at launching 3 major clinical programs in AML, small cell lung cancer, and in combination with doxorubicin as a cardioprotective anti-cancer drug. On top of all of that, with the opportunity for composition of matter patent protection over the drug that we have. So really quite an amazing journey, and it still has quite a long way to go. So I want to just touch a little bit on the background and refresh some of the things that we've been through and really provide context for when I hand over to Rodney, where he'll take you through the non-small cell lung cancer program, who has been instrumental in getting us to this point. So just provide a little bit of background for those of you who remember the talk a few weeks back now about mechanism of action. The thing that we've really made big strides in the last year, or actually even less than a year since the beginning of the year, is working out how bisantrene actually works. It wasn't, as everyone thought, as a traditional chemo agent. It's a specific targeted agent that binds to a particular structure in DNA and RNA called G quadruplexes. Ultimately, no investor really needs to know the details of all of this. It's very, very complicated, What you do need to know is that it controls a whole series of very important cancer genes, none more important than MYC, which is really the sort of the key master on-off switch for cell growth in the cell. It also has a series of effect on other pathways, which will be very important for EGFR, small cell lung cancer, and it also has an effect on M6A levels in RNA. So all of these things have come together in bringing an understanding to what were initially a whole series of, we know bisantrene does this, we know bisantrene does that, but we had no real understanding about how it managed to do all these different things. We now know, and this really allows us to target the right opportunities. And none more exciting than MYC. This has been a target, a protein that the pharma industry has been trying to develop drugs for the last 40 years without success. The reason why is the protein itself is unstructured. You really can't find a drug that binds to it very easily. And so as a consequence of that, there are no MYC drugs in development in any pharma pipeline. This is despite its key role in an enormous number of different cancers. Over 70% of all cancers have dysregulated MYC activity. That amounts to more than 1 million cancers in the USA alone, several million worldwide, meaning that the potential market for this is extremely large. Best way to do this is jump on your favorite AI and ask it, how much would a generalized MYC inhibitor be worth in sales every year? And you'll get some extraordinary numbers. Whether they're true or not, I don't know. It doesn't really matter too much at this point. It's a very, very large market and a very, very large opportunity. Another very large market are the tyrosine kinase inhibitor market. So that's around about 55 billion US dollars a year at the moment, and it's continuing to grow, expected to double over the next 8 years. And the reason why is that these tyrosine kinase inhibitors regulate the genes and proteins that are very important for driving cellular growth. And as a consequence of that, many cancers have mutations in tyrosine kinases. There's about 660 or something like that, different tyrosine kinases in a human cell, different ones for different cells. So there's a lot of different targets, and these particular inhibitors are a major, major market, existing market out there. And they can be quite valuable, even though this is not really these targeted agents, and they can be really quite valuable very early on. So this is an example here of Mirati Therapeutics. It developed a drug that could target KRAS. So KRAS is an important growth regulating protein that is often mutated in cancer cells. Previously, it had been considered undruggable, much like MYC. But Mirati managed to work out they could target a specific mutation that's found in a relatively small percentage of mutated KRAS cancers. The consequence of that is even before they got to approval, they got to a 12 billion US dollar market cap in early 2022. They unfortunately were beaten to market by Amgen, which developed a similar type of drug. But ultimately, at the end, even after failing, I guess, if you want to consider it that way, they still ended up with, more than $5 billion acquisition from BMS off relatively small sales numbers. So BMS reported only $123 million of sales of Krazati in 2024. It's growing rapidly, but it's still a relatively small market, yet they pay large pharma for a targeted agent that is unique. You can get a very large multiple off the existing sales. So it's just an interesting example there. But I just wanted to, before I hand over to Rodney, just take you through the process that we went to end up with the programs that we have. Obviously, AML is more obvious given the long history Race has in AML, and with the opportunity to go into phase 3, we thought that this would be an excellent opportunity to get the drug onto the market as quickly as possible, even if the opportunity is not as large as it might be for other indications. But we wanted to really take the mechanism of action that we discovered earlier this year and sit down and work out where we could best use it. Because we're able to target MYC, we have the ability to actually target a wide range of different cancers. So we sat down and went through with a systematic checklist of properties that we wanted to make sure that we had, so that we identified what is going to be the indication to go after that gives us the best commercial potential from this. This is an opportunity that not many biotechs or pharmaceutical companies have. For most drugs, you're limited to a relatively small range of different cancers. But for us, because of this MYC targeting opportunity, we have the ability to pretty much go after any cancer that we would want. So we decided to be really rigorous, strict, and really focus in and try and identify what would be the best opportunity out of all the possible opportunities that exist within the oncology market. The first thing we looked at, it had to be a large market. We wanted more than $10 billion a year in sales potential there. So we threw out all the orphan indications, all the smaller cancers, and really honed in. Obviously, lung cancer is the most common cancer that exists. So we pretty quickly focused in on lung cancer. We wanted to have really excellent and supportive preclinical and clinical data. We wanted to have a pretty good idea that this was going to work. If we began a clinical program, what's the probability of success? We want to make it as high as possible, so it's much more likely to generate a return. We really wanted to make sure that we're solving a pressing unmet clinical need. There's no point being the sixth or seventh drug in a class treating a cancer that's well treated by other treatments. It needs to be solving something that all clinicians are crying out for, patients are crying out for, so that we can make sure that we're actually solving a real problem. Ideally, we wanted there to be lots of pharma players in the space, so we have many potential partners that we could partner with to develop this drug. So some spaces are dominated by one player. There are no other competing pharma players that have competing drugs on the market. And as a consequence of that, you're really dealing with just one company if you want to partner. And that obviously just reduces the probability of not only partnering, but getting a good partnership deal. The other one, we wanted to make sure that it could be combined with a proprietary drug. So we had a pharmaceutical partner that we could actually partner with their drug. So that moves hands in hand with having many players. Obviously, the trial, we're a small company relative to certainly the large pharmaceutical companies. So we needed to have a trial that could be done both rapidly and low cost and generate the proof of concept data that would really attract interest from any potential partner. We want to make sure we had a large effect size, so we don't need very many patients. You don't want to be running some trials. You need thousands and thousands of patients to know whether your drug works. In some indications, you only need a handful of patients to know whether your drug works. So they have large effect sizes, and they're obviously very attractive. The other one we really wanted to focus on is the FDA likely to give accelerated approval? So not all oncology drugs have to go all the way to phase 3 or 2 phase 3 trials to get approval. Some are approved as early as phase 1B. And so we wanted to make sure that we're in an indication where that was, a possibility, or at least a good possibility. And the other one too is if you wanted to extract maximum value, you want to have something that can be a template. You can apply the same basic principles to one indication, you can take it and apply it to other indications. And you can see this as being label extension. And Big Pharma is particularly interested in this. We can think about something like Keytruda. It was originally developed and approved for melanoma, but over the years, Merck has taken that and run multiple trials in different indications and expanded the range in which the drug can be used and built it up into a $25 billion a year blockbuster. The outcome of all of this is we identified EGFR mutant non-small cell lung cancer, which is a very interesting type of lung cancer, and which Rodney will tell you all about.
Thanks, Daniel. Just want to say thanks to everyone who's attending the webinar for your time. And also appreciate the opportunity to present on the lung program, which hopefully you'll see is definitely very attractive, I think, for the company to pursue. So, like most of these presentations, we'll start off with some background statistics in terms of lung cancer. Probably no surprise to anyone that it's one of the major types of cancer which people can suffer from. Certainly last year, looking at something around 2.5 million global cases, with a significant number of those in the US, but also a far larger amount in China. So that's what we're sort of seeing in this new age, I suppose, with China having come on board many years, quite a few years ago now. They're quite a large player in this space. Unfortunately, with lung cancer, it's still one of the leading causes of mortality, still looking at something like nearly 2 million deaths annually in the coming years. So definitely a massive unmet need. As you're probably aware, smoking is one of the primary causes of lung cancer. But it's also, I think, important just to realize too that there's a large proportion, around 25% of people who've never smoked or don't have smoking as the cause, and that's just from down to environmental factors as well. So altogether, definitely a very tragic disease and one that we think we can make a real dent in. I guess just quickly too on that. The lung cancer is broken down into two main types. One is small cell lung cancer, which is the junior partner, but by far and away the larger one, around 85% of cancers are non-small cell lung cancer, where they're lung cancer. And there's some other histologies under there, but we don't need to go into. But certainly out of that large proportion of lung cancer, non-small cell lung cancer is the key one to be looking at. Thanks, Daniel. And why are we interested in epidermal growth factor receptor mutated non-small cell lung cancer? Well, EGFR was one of the mutations that was identified early on in research, and for which a large number of tyrosine kinase inhibitors, which is the class of drug which is targeted against them, were developed. So through all the work that's been done, we know that EGFR is one of the main drivers of cell growth. Once it becomes mutated, that cell growth becomes uncontrollable, the cells are off the leash, and you get runaway lung cancer. Importantly, there are two main types of mutations or regions which are mutated in the EGFR gene, and that's well documented, and inhibitors have been developed against those. So that's all well established, leading to the fact that it's a $10 billion market for us to be getting into. Thanks, Daniel. There is a little bit of a difference between populations around the world in terms of how many patients have the EGFR mutation. So I guess the general premise is that in European populations, and including North America, the incidence is around about sort of 15% across those regions. But importantly, and as again coming back to the idea of Asia and China in particular, we see that it's more than double, if not triple, those levels in those populations. So it's definitely a huge unmet need. And it's also a main driver of a lot of activity in China and Japan in terms of developing their own tyrosine kinase inhibitors to address the problem in their populations. So again, a lot of interest. Probably the next slide would be good, please, Daniel. An important factor, there's certainly a question around whether immunotherapies could be used in the space, and they have been tried. Unfortunately, where there's mutation in the EGFR receptor, those immunotherapies, for whatever reason, do very, very poorly. So what this means is we're unlikely to see immune checkpoint inhibitors flooding the market for EGFR mutation, and the burden of treating the disease still rests with tyrosine kinase inhibitors. For that reason, we're already up to the third generation of these drugs. Basically, the reason for that, of course, is that they're actually working rather effectively, particularly initially in patients. So most people respond well to at least one of those generations of inhibitors. But unfortunately, well, I guess as a consequence, we've got that large market and a lot of activity in terms of, what do we look at? 14 approved tyrosine kinase inhibitors across the first, second, and third generations. That's in the USA and China and around the world, and quite a few more still in development in late-stage clinical studies. The shining light in this space is osimertinib, brand name Tagrisso. which alone last year had nearly $7 billion in revenue coming in for AstraZeneca. And we certainly know that even though it's a competitive space, AstraZeneca in particular are fighting very hard to hold on to their lead. I think as you know, by way of reference, that nearly $7 billion of sales represented about 13 to 14% of AstraZeneca's entire revenue across all their businesses. So definitely something they're trying to hold on to. So we've got intense competition, and the team have put together this table. I think maybe Daniel was instrumental in this, but it just highlights again the competition that's there, the number of players trying to get into this space. You've got osimertinib there from AstraZeneca as the standout, but even coming down the list there, you can see a number of other third-generation inhibitors coming out of China, and they're still looking at revenues of $500 million already for a number of those. We've highlighted lazertinib down there towards the bottom. This is a more recent entry in the third-generation inhibitor space. It's being brought through by Janssen, who are a subsidiary of Johnson & Johnson, one of the largest pharma companies in the world. And just an example of even how someone lagging way behind can still get some decent sales. You can see there that in Q1 this year alone, $141 million from very minimal clinical evidence, just a couple of studies that gained them approvals, and already they're starting to gain some traction in the space. So it basically, and the next slide as well just talks about some of the clinical trials that are going on. And you can see there that people are already starting to talk about fourth generation inhibitors. So it's still a very active space. We tend to think that the fourth generations probably aren't going to get or go as well as the third generations have. They're looking at a particular type of mutation in the EGFR, which is a minor reason for resistance. So we're kind of already seeing some of them dropping out of the play for business reasons, but still very active space. So this one's a little bit complicated, I guess, in terms of what it's showing. But if I just draw your attention to the green line, there's actually two of them there. And what this is showing is that osimertinib, the market leader, and lazertinib, that one that we just spoke about there in bold, basically perform the same. So patients, this is showing the number of patients with progression-free survival, meaning their disease hasn't progressed beyond a certain amount. And you can see that about half of patients are still doing well at about 15, well, probably 16 to 18 months there. So no real difference between them, and that's kind of what we think what will happen with the third generation inhibitors. So probably osimertinib from AstraZeneca is still going to maintain their lead as the most recognized brand, but other ones are coming through, adding to the competition. I guess the other important thing to take away from this is that although patients are doing well out to about 18 months, there's still this steady decline. Patients are still failing all the way through. So for those patients who've already progressed, they definitely need something to help them out. And this is part of the reason why we think we have a play in this space. Even with the fourth generation inhibitors, targeting a single gene is just a bit of a failed strategy, we think. The problem you've got is that cancer can find a way around. They can bypass, inhibition in one particular receptor by activating other receptors. So here you can see in blue, you're hitting EGFR, that's fantastic. But over time, cancer cells will start to mutate around other receptors to make sure they keep driving their cell growth and they can come back, basically. So, and there are inhibitors known for some of these other ones, like HER2, the Herceptin, for instance. But again, knocking off that one may still leave many other ones available for cancer to try and work around as well. So the real problem here is that even within one patient, you still have multiple bypass mechanisms, multiple mutations kicking in, and there's always this issue of subclones. The idea there is, if we look at the figure on label A here, you may start off with a normal cell, which mutates and becomes cancerous. So that's that red filled in circle. Noting that there is a particular mutation in EGFR. Already, though, even at that point, there's already another mutation sitting there underneath. And then from that point onwards, this patient was monitored and retested over time. And you can see that from that red circle, we now have the blue circles, they go out to the yellow and the greens and so forth. And you get this proliferation in lots of different mutations. And these are all different cells starting to grow out from underneath the original EGFR mutation. So whilst that might be being controlled, all these other cells are now starting to grow and proliferate from underneath, and eventually, unfortunately, patients progress. And you can see that again with some of the other patients here, patient B, patient E, patient F. Different mutations, not always the same ones, but lots of them starting to grow through. So the problem there is using a targeted therapy to take out just one of these mutations isn't really going to help these people with all the other ones coming through. We really need a treatment that can mop up or restrict the growth of all of these subclones. And that's what we think we can do with EE bisantrene. Look, the idea is not exactly novel, applying a new agent on top of an existing therapy, and we acknowledge that others have started doing this, of course, and this is what happens in cancer. So Johnson & Johnson, the makers of lazertinib, ran the Mariposa study on the left-hand side there, where they added in it's a bispecific antibody targeting a couple of other pathways. And indeed, they did see an improvement. So you can see that the blue line leads to longer progression-free survival in those patients. Again, as we've highlighted, though, patients are still progressing steadily through them. So that, again, a number of those patients have dropped off. They need something else. Interestingly, too, more recently, just this year, we had publication of FLORA-2, which was a study run by AstraZeneca, where they looked at adding chemo on top of osimertinib. Again, this idea of trying to pick off all of these different subclones and try and get some benefit to the patients. So here, yes, indeed, they were able, in the orange line, to delay somewhat the progression of their patients. Again, though, there are still patients falling through the cracks all the way along and will continue to as well. Inevitably, patients will fail on TKI treatment, and they definitely need something coming through in the end. There's also another challenge, I think, with the use of chemo in terms of platinum. It's relatively effective, but it does come with additional toxicities. So this particular study, patients only received 4 cycles, and then after that, they just were left on a bit of pemetrexed and osimertinib. So what we're seeing is, in conversation with clinicians, there's this debate going on at the moment around whether it's actually better to hold back platinum for the end and just keep people on osimertinib and then try and treat people later. So in the community out there, there's a bit of a debate as to what's the best approach, because you only get a limited use of these chemotherapies before patients can't tolerate them and they actually stop working as well. So there's a few things there that we've identified that, we feel EE bisantrene can address. Thanks, Daniel. And so what are those things? So we've obviously identified the bypass signaling. We want something that can knock off all these other ones. Where does EE bisantrene fit there? Well, the nice thing is MIC-MAX. So this MIC protein, as Daniel spoke about, as you can see in the figure, it sits downstream of a lot of other these growth factor receptors as well. So even though they may be being mutated, we can perhaps intercept them through MIC and our activity in downregulating that and stop further cell growth. Obviously, we want to be able to target different subclones. Again, this particular mechanism of action that we've identified can assist in that. The other important fact is, based on the clinical history with EE bisantrene, it seems that patients will tolerate it well almost indefinitely. So we know patients in earlier trials with EE bisantrene went up to 17 cycles, so a couple of years of treatment with EE bisantrene. And that's really good, because if patients are getting benefit, they want to stay on a drug that's working for them. So you can start to turn this disease into something that's more chronic rather than a death sentence. And that compares and contrasts with other agents. As I said, the platinum drugs have limitations in terms of how long patients can stay on them and tolerate the treatment, but also other ones like doxorubicin. We know doctors are limited in terms of how much they can give and for how long before patients have to go on to other therapies. So something like EE bisantrene really positions us really well to be able to address this issue for patients and keep them on treatment for longer. Which dovetail nicely with patient quality of life, of course. And we know, EE bisantrene has some fantastic features that patients really hopefully will like. Things like lack of hair falling out and obviously, lack of effect on patients' hearts as well. So that'll be quite important, particularly in an elderly population. And the other important one, of course, is really need something that's proprietary and has patent protection, which we now have for eibuzantrine. So always the challenge with these things, there's lots of publications around generic drugs and combinations and so forth that seem to work okay in animal models, maybe early phase clinical trials, but very rare that anyone's going to stump up $100 million plus in order to take them all the way through onto the market. So, because ultimately, if they try and do that, there's a generics competitor waiting to undercut them, and they risk not recouping any of their costs. So, with all that said, historically, when we were looking at this last year, we were very much focusing on the topoisomerase 2 alpha properties of bizantrine as the way forward to address this broad targeting that we wanted to go after, in addition to the mix inhibition. And what got us really excited was a publication out of Emory University over in the United States, happened around about March last year. And what they reported was something really interesting. So when they developed cells that had resistance to osimertinib, they noted that addition of a topoisomerase 2 inhibitor was actually effective at overcoming that resistance and restoring the cell killing. So this was fantastic news from our point of view, where there looked to be some very good synergy there. And we know that eibuzantrine is absolutely a topoisomerase 2 alpha inhibitor as part of its mechanism of action. So this really lit a spark under us. So we delved deeper. And you can see from the figures here, what we're looking at on the left-hand side is PC9. It's a type of non-small cell lung cancer cell. AR means acquired resistance. So this is one that they have been treating with osimertinib and created a resistant cell line for. And the really nice thing about the study was they didn't just look at topoisomerase 2 inhibitors like doxorubicin or etoposide, which is the orange and the red, but they also looked across the board at other chemotherapies, so paclitaxel, cisplatin, gemcitabine, cyclophosphamide, all these other traditional chemotherapies. And they saw nothing with those. So there's no movement in PC9, but also HCC827. Only the topoisomerase 2 inhibitors moved the dial there, and in a significant way. They then looked at some patient samples that they'd got from trials, and they noted that topoisomerase 2 in patients who were resistant to osimertinib treatment and other TKIs was upregulated in about 2/3 of those patients. So again, a very clear marker that the majority of patients are in the situation where a topo 2 inhibitor could be effective. The icing on the cake was when they went and looked at an animal model. So PDX stands for patient-derived xenograft. Essentially, you take a tumor out of a patient, you split it up, you put it into some mice, and you start treating those mice and see what happens. So in this case, the grey line there you can see in those three figures is vehicle control, just saline treatment. So you wouldn't expect anything to happen. That's essentially the tumors growing out. The blue line is treatment with osimertinib. So although there's some delay or reduction in the growth rate, you can see that those tumors are still growing out, certainly in the top two, which kind of goes with the fact that these patients were starting to get resistant to osimertinib treatment. Etoposide is VP16. You can see in the green line there, it had some effect as well, delaying the growth, but still the tumors continued on their merry way eventually. But the really exciting thing was the combination. So put the two together and you basically eliminate those tumors. They go to zero. So it's very exciting. Again, these are patient tumors, so much more real world. In the bottom figure two, the scientists continued treating with osimertinib. You can see that they're going out nearly to 6 months there, at which point they saw eventual resistance naturally developing even further in this patient. In this model. So at that time, they added in etoposide on top of the osimertinib, and they were able to then rapidly decrease the size of those tumors. So showing that even after a patient potentially could be on osimertinib for a period of time, you can come in with a topoisomerase 2 inhibitor and knock down the tumor effectively. So all of that was really, genuinely exciting for the preclinical team. We got energized around this and thought this looks like a fantastic thing. Again, while the researchers here used etoposide and doxorubicin, it's really hard for anyone to be developing those as drugs in this space. So we feel it's great proof of concept, but certainly the field is calling out for eibuzantrine to come in. Thanks, Daniel. So touching again, I won't labour the point, but just to reiterate, a number of the mechanisms of actions that are result from the G quadruplex binding properties of E-bisantrene are exactly what we're looking for. So the mixed silencing, and notably at concentrations there which are highly clinically relevant. So we expect to see concentrations of the drug at those levels in patients, which is great. M6A levels increase with treatment, and again, we know it's been reported and we've confirmed topo 2 activity of E-bisantrene. So we're ticking a lot of boxes there. We then took the action of reaching out to those researchers at Emory and just getting them to drop E-bisantrene into their tubes and have a look and see what happens. So fortunately, well, happily, you can see from the top there in that PC9 and the HCC827 model, which again were resistant to osimertinib, which is confirmed with the yellow line there. You can see treatment with osimertinib really doesn't do anything at these concentrations. But when you put E-bisantrene in, you get the red line. So that separation indicates that more cells are dying at those concentrations than what you would get with each agent on its own. So again, we're looking at minimal additivity, if not synergy, in these models with the combination with E-bisantrene, which is fantastic. We commissioned some animal studies ourselves. This time, the bottom there, you can see we've given osimertinib and then the combination with RC220, which is our formulation of E-bisantrene. This was done in a PC9 lung model where the cells are actually sensitive to osimertinib. So what we were doing here is pressing the cells, and then at a certain point at 28 days, we took away the drugs, and we were just seeing what the effect was and how quickly the residual tumor cells would grow out from underneath. So you can see there in the red, osimertinib, the cells are growing away after the osimertinib has been taken away. But pleasingly, the addition of RC220, E-bisantrene, led to a significant delay in that regrowth, indicating that there's a combination of suppression of growth, if not cell killing there going on with the combination, which is exactly what we want to see. And for those inclined, there's some information there around apoptosis. That's just a particular pathway that cells use to die. It's a programmed cell death pathway. And that's just indicating that that indeed is a major pathway for the combination of E-bisantrene and osimertinib. So around 50% of those cells in the PC9 model and about 30% in the HC827 model are dying via apoptosis, compared to very minimal for osimertinib treatment alone and comparable to doxorubicin. So again, really looking like a good play for us. So again, just summarizing, when you've got resistance with osimertinib, you see on the column on the left, these are the properties. So you get an increase in topoisomerase 2 alpha activity. We know E-bisantrene can inhibit that. So that's a big tick. We also know with resistance, you get bypass signaling. So you want some common mechanism in order to head that off. And we know with E-bisantrene, we can silence MYC and also other oncogenic expression as well. So there are other pathways there which we also inhibit too, things like telomerase as well, which are great and will be important. And finally, also with osimertinib resistance, there's a decrease in M6A RNA levels, which Daniel's already pointed out. And we know that E-bisantrene can increase those levels and counteract that. So again, we're ticking a lot of boxes with bisantrene here. Thank you, Daniel. So with all that behind us, compelling market, clinical unmet need, really compelling preclinical data to support a move into the clinic, the team have gone ahead and started pulling together things like the protocols and submit to ethics and getting everything ready, basically, such that we can start this program as soon as we're able to. So we've called this study the Harness 1 trial. You can see what it says there: halt resistance in non-small cell lung cancer with EGFR sensitizing mutations. Importantly, it's a phase 1 study, which basically means that we're looking at the safety of the combination. We have to do that first. So we've got to establish what dose levels we can give to patients on top of osimertinib. So you have to go back to phase 1. The goal here is to identify a very robust maximum tolerated dose. So we have confidence in taking that forward into future clinical trials. We're also going to be looking at selecting patients for the study using circulating tumor DNA, ctDNA there, and I'll talk about that briefly in a minute. But It enables us to identify patients before they've actually had progression by imaging, and may enable us to access them ahead of other competing trials. We think this could actually be quite an advantage for us. But basically, those patients will come in when they become eligible based on a positive result, and we'll also track patients with ctDNA through the trial. The idea there is it will give us an early read on how they're responding to treatment, but we'll touch a bit on that in a moment. Other important factors for the study, we'll be looking at, because we're in patients, we will be looking at effects on tumours and survival. So objective response rate, progression-free survival, overall survival, these sorts of metrics that you would commonly do, in addition to the ctDNA monitoring. Looking at, the moment, we're looking at 5 sites, all in Australia. So a couple in Sydney, a couple in Melbourne, 1 in Brisbane. We're already quite advanced with discussions with a number of those sites. In fact, ethics has gone through down in Victoria. So we're in that process of working through responses and the like. So that's really positive. In terms of patients for the study, look, we estimate around about 100 patients we needed on screening. We know from the literature that about 5% of patients in any given month will progress by ctDNA. So we kind of need a pool of patients in order for enough of them to be dropping into the treatment phase of the study at any given time. For the phase 1A, we are terming that the dose escalation, where we're looking at increasing doses of RC220 on top of osimertinib. It's hard to pin down exactly how many patients we need. We think 40 would be an upper limit, but the study is going to be using Bayesian statistics in order to kind of home in on the optimal dose, the maximum tolerated dose of the drug. So it's adaptive in design, but the key is we get a really good measure and confidence in the maximum tolerated dose that we can then take through into the expansion phase 1B study and fully characterize. That second part of the study, the phase 1B, will also bring through a second dose level, and that's to make sure that we've got the right MTD. So we'll have a lower dose level. If the MTD turns out not to be the MTD, we've got a lower one to fall back on. It also addresses regulatory requirements. So we're aware that FDA has a Project Optimus program running, where they are very much encouraging, shall we say, companies to carefully characterize the doses that they're bringing forward into larger studies. So they really want companies to fully justify the dose they take forward and have done sufficient characterization in order to achieve that. And so basically ticking that box means we'll hopefully have a smoother pathway with FDA in those discussions. Estimate the cost for the study at around about $11.5 million before R&D tax credits come back in. And in terms of recruitment, looking at about 2 years to get the study done. Patients on study will be treated for up to 1 year. So just be aware as well that 2 years to recruit, but also any patient towards the end there will still be on for another year at the end. So just be aware of that. But look, with all the work we've done, the team at Race have really been pulling out all stops, and our vendors that we've engaged with already have been fantastic. We've got a long way towards being ready to go. And really, with all the approvals being granted as we expect, it's certainly no risk, I think, of us being unable to start in Q1 next year. So really good support, and we're in a good situation there. So yeah, it's just touching back on circulating tumor DNA and what it actually means. So the concept is that if you have a tumor, you've got lots of cancer cells there that just through their natural cycle will die. Now, when they die, the cell membranes lyse and they release their contents. And so you'll get fragments of their DNA naturally leaking out of the tumor and into a patient's bloodstream. So what this provides is a proportionality with that tumor, and we can actually use modern techniques to measure the levels of that DNA. Now, as the tumor grows and gets larger, there'll be more cells there, more cells dying, more DNA leaking into the bloodstream. So it provides a good way of, I guess, initially tracking the progress of that patient's disease without having to resort to imaging. And this is becoming more and more accepted in the oncology community, and there's lots more clinical trials now up and running looking at and using ctDNA. But what it also means is that you could imagine if you have a successful treatment, you kill off a lot of those cells, you get maybe a spike in ctDNA from all the DNA flooding the system, but it'll be washed out eventually. And what you're looking for is, in a successful treatment, the levels of the DNA at the end in terms of where they were relative to baseline should be a lot lower. So again, you can actually track how well that patient's responding to treatment, and then you can continue tracking to see if the levels come back up or stay down as well. And that's what we'll be looking to do here. So it provides a really non-invasive way and of tracking patients, and something you can do far more often than imaging as well. If you're exposing patients to radiation, there's limitations there, whereas this, we can be sampling patients very regularly throughout the study and keeping track of how things are going for them. So here, thanks, Daniel, simplified schematic, more capturing what I've just described. So we'll have a screening cohort where we're looking at patients. When they become eligible on account of a positive ctDNA result, they drop into one of those two stages of the study. Phase 1A first. Once that's finished, then they'll drop into phase 1B. You can see the 2 dose levels that we'll be evaluating. And then the idea is to, through all of this, we pull out one optimal dose that we know is going to work, and we send that on to the more pivotal studies that regulators will need. We won't gloss over this too much. Well, we'll gloss over it a little bit. But as Daniel highlighted at the start of the talk, we've got a lot of boxes that we think we can tick, that we would like ticked, that we think we can address. And fortunately for us, non-small cell lung cancer with patients with EGFR mutations ticks all these boxes. I mean, it's just a great space to be getting into, and I think we can make a really meaningful difference to patients and add a lot of value to the company. Thanks, Daniel.
Thank you, Rodney. Excellent discussion there. So I will take out the last bit, which is the AML phase 3 program, and try and really give an explanation about how this has come about and why we're undertaking this program. So many of you will remember that RC110, which is the formulation that we developed as close as we could match to the original Ledley formulation from the 1970s and 80s. And we trialed that in 2 trials in Israel, where we saw both on the first trial was as a mono agent, just on bisantrene on its own, and the second trial was in combination with 2 nucleoside analogues. And both of those trials, 40% response rate, excellent outcome in what were extremely difficult to treat patients. It's really hard to overestimate how difficult these patients were to treat. It's just the nature of the patients that were available, but the real need that all these patients were, weeks from death if they didn't respond. So as many of you know the stories about this, it's a really great opportunity, really positive results. And, we've been looking around, how can we take this forward? Initially, we thought, okay, maybe we can look at an IST here in Australia. And that's still certainly something that's very attractive and become more attractive as we now know the mechanism of action, particularly in acute myeloid leukemia, where about 90% of acute myeloid leukemia cancers overexpress MYC. MYC is extremely important in AML. So, we've got something pretty exciting for this area. And this is sort of reflected in, how is bisantrene done compared to other drugs that have been either standard of care, things like cytarabine, but some of these newer targeted agents that have come through. One of the big problems with targeted agents is while they can work better in the patients that happen to have the particular mutation that the drug is targeting, can mean that there are very few patients that actually have that mutation. So some of these drugs target, less than 5% of AML patients. AML is already an orphan indication, so you can be talking literally down to hundreds of patients worldwide will have the appropriate drug. And that's great if you happen to have that mutation. But if you don't, which, more than half the AML patients don't have a targetable mutation, what do? You rely on either traditional chemotherapy or something like bisantrene, which is not particularly targeted to any particular molecular target because of this broad efficacy across a wide range because of the G quadruplex binding. Of course, the really big question is, that's great. How can we actually go about doing a trial? Phase 3 trials typically are not cheap to undertake. It's not uncommon to spend $1 billion in a phase 3 program. If you have to run 2 phase 3 programs of a couple of thousand patients in each trial, you will not have much left after spending $1 billion US. Unfortunately, or maybe fortunately for the shareholders, we don't have access to $1 billion. So what can we do within the constraints that we have, and is there a pathway forward? And until recently, we didn't really have a very good, cheap pathway forward, but we were looking around, or certainly I was looking around at, what some of the other companies in the space, what are they doing? And I came across the Moleculin's trial design at the beginning of this year. They put out an announcement. They're a company based in Texas developing a anthracycline derivative called animicin. So it's like doxorubicin. And they went to the FDA after they ran a couple of phase 2 trials and said, this is what we want to do for phase 3. And the FDA, at the beginning of this year, gave their approval to a design that looks something like this. And I looked at this and I thought, wow, we could do exactly the same thing with bisantrene. We've got all the same supporting data. We have a drug that's more effective than animicin in these patients, and we could go straight into phase 3. The cost of this is relatively modest and relatively fast. And I thought, this is actually something worth studying. So this led to a new plan, which we've been working on for some time, utilizing both a combination of pathways, parallel pathways, one pathway designed to get the drug approved for AML as quickly as possible. This will get the drug on the market, give us control over our destiny. One of the good things about we've got great opportunities in lung cancer and cardioprotection anti-cancer. Those are very, very large opportunities, and they're beyond the ability of Race to control its destiny in those spaces. In an orphan drug indication, while the dollars are not as high, the potential dollars anyway, the market is something that a small company can actually do. And I think we've seen this happen on the Australian market. Neuron's a really good example of this. They can take, for an orphan indication, take it all the way through to commercialization and be generating revenue from that and support other programs after that. But at the same time, we wanted to make sure that there's a lot of change going on in the AML space, clinical space. We want to make sure that bisantrene's got an opportunity to be used in an optimal way, but that doesn't have to be the same as the approval way. So we can take what we've done in the past to get the drug approved, and we can in parallel run a parallel path to work out actually what's the best way of using this drug. The two don't necessarily have to be together. That can really cut down the costs of such a trial. So this gives us the strategy for how we can get there. There's a few things we have to do. First one is we don't really want to take RC110 all the way through to approval. RC110 has to be delivered by a central line, which isn't a problem in AML, but it is for everything else. But it would mean that we would have to have 2 what are called CMC, or chemistry manufacturing control programs. They typically cost between $10 to $50 million to get your drug all the way through to approval, depending on the drug. It's obviously with a drug like bisantrene to be on the lower end, but that's even at $10 million. It's $10 million I would like to prefer not to spend if I don't have to. And the other one is we hadn't actually, Project Optimus came in only recently. It's a requirement these days that you generate multiple dose levels for any new oncology drug. So it wasn't done initially because it wasn't a requirement, but now it is. So what we can do is we can split the phase 3 trial into 2 parts. The first part basically involves a bridging study. It takes advantage of something that was built in originally right from the beginning with the development of RC220, and in fact was probably the most difficult thing to overcome in the development of RC220 was to make it identical in its pharmacokinetics and pharmacodynamics to the original RC110. When you reformulate a drug, you almost always end up changing those 2 properties. It's very hard to come up with different formulations that have identical PK and PD properties. But we were able to accomplish that. after quite a lot of effort. But that gives us a huge advantage now. We can now bridge across, do a relatively small study, 40 patients in total, and bridge across RC110 to RC220, which would allow us to take RC220 through to approval for AML. Second thing we can do is we can compare two different dose levels. In this case, 4 days versus 7 days of continuous dosing to work out what's the best dosing level and satisfy Project Optimus requirements. Accomplish all of those things, have a meeting with the FDA, and then move forward with the optimal dose, either with 110 if we can't show the bridging's done, but with 220 optimally with the correct optimal dose level, and then expand that out in an efficacy study. This brings us to what would give us FDA and EMA approval. You only need in orphan drugs 1 phase 3 trial to get to approval. So this is something that's within our control of being able to take bisantrene all the way through to approval. This is just an example here of maybe what you might do to try and work out the optimal. It's just an example of using it in combination with venetoclax, but of course RC220 can be used in combination with many different drugs. And it's certainly something we've just found in the past that it works really well with a range of different AML drugs. Which one is the optimal one to take forward would be something, and maybe multiple studies. All of those things are something to establish down the road once we get further down the process of the phase 3 trial. So why would you want to go about doing this? Well, it provides a rapid path to approval. Ultimately, if we get our drug approved, it has value, even though AML is a relatively small market. Much like Neuron, a small market can generate a very large number of sales. And the reason for that is you can charge a very large amount for an orphan drug indication. And some of these AML drugs that have come to market are priced at, $450,000 per patient, per treatment cycle, not one cycle, but series of cycles. So you can charge an awful lot for a very small patient. Really builds on a model we know has been agreed by the FDA in recent times. So we're not just flying blind. And for the other programs, it really provides us with compelling preclinical data around MYC. Because we know AML overexpresses MYC, and because we can sample the cancer cells out of the blood very easily, we can demonstrate time course inhibition of MYC by bisantrene in a patient. And that's incredibly valuable data to show any potential partner or acquirer that you can say, oh, yes, we actually are in patients targeting MYC. We can get that from the first patient that we treat in this trial. Opens up the opportunity for a pediatric PRV. We can add a sub-study onto the expansion stage, put pediatric patients under 18. You need a very small number of those patients, typically 12 to 24 patients is enough to get you approval for pediatric indication, and that comes within a pediatric PRV. So that they're worth on the order of $100 million to $150 million each if you can get one. Overall, the whole thing is designed to be capital efficient. Can we do this within the resources we have? We have this $25 million coming up with the piggyback options. A number of those have already been converted, which is fantastic to see. But can we, within that resources that we have, can we actually undertake this trial? So we can undertake the first stage, the bridging Project Optimus stage, but we haven't yet have the resources for the dose expansion, but that's certainly, we won't be doing that for a little while. And the same thing with the Australian IST. It's very, very cheap, $4 million for 60 patients before the R&D tax. So you're looking at only a little bit over $2 million to run a proof of concept trial that will prove out which one it is. So getting to the last slide, we've announced, 2 new clinical trials on top of, what is still a major focus of RACE, which is the anti-cancer cardioprotection. Nothing's changed there other than we have a better understanding about why that works, de-risk from that perspective. There's still a massive opportunity. We're still continuing with that. No attention or resources have been diverted, but it gives us 2 new opportunities that we can go forward with right now. Rather than having that money coming in from the piggyback options just sitting in the bank collecting interest, we can actually get into the clinic, develop some of these programs and generate real value for our shareholders. So the phase 3 program in AML and this phase 1A/B in EGFR lung cancer. Everything can be done. Obviously, we've had 18% converted already. I suspect that number is already up. That was from the 14th of November. And I'm sure as everyone remembers, I own 12% of those options. I'm very much motivated to see these programs go forward. Planning for all these activities is underway. As Rodney has pointed out, he's done an amazing job, leading the team to getting the EGFR. It's all ready to go as soon as we have the resources, but we're not going to go launch into a program that we can't finish. And with that, I'd just like to thank all our shareholders for their continued support and enthusiasm. We wouldn't have got to where we are today without your support and enthusiasm. From my perspective, it's all of our company. It's not just management's. It's we're all in this together, and if we succeed or fail, we'll succeed and fail together. So thank you.
Thank you, Daniel, and thanks, Rodney, for your presentation. That was terrific. I'm conscious we've taken people the whole way through to 2 o'clock across your lunch times. We're about to open up the floor to Q&A, but please, if you need to drop off, feel free to just slide out because it's I know it's the middle of your work day, but thank you very much to anyone who's already dropped questions in. If you'd like to do that, you can use the Q&A button in the bottom of your screen. So straight into those. Is everything that shareholders have painstakingly learnt about FTO now useless? Daniel.
Well, everything I've learnt about FTO is useless. No, the answer to that is no. In fact, bisantrene acts as though it is an FTO inhibitor. It causes a rise in M6A levels, and that's what actually has all the effect in the cell. It's not the inhibiting of FTO that's important. It's the rise in M6A levels in the RNA that actually drives all the biological activity. Bisantrene causes that rise. It just does it in a different way. Instead of inhibiting FTO, it has a more direct effect that causes the M6A levels and has all the effects that causing M6A levels to go up have in a cell. So that's really important. It's important for AML. It's important for EGFR lung cancer. It's important in solid tumors. Everything that you've learnt about FTO transfers straight over to the new mechanism of action. So, yeah, I've spent, I don't know how many hundreds of hours learning about FTO and M6A, and I don't feel like I've wasted my time on it.
I can feel Alan, who asked that question, breathing a sigh of relief. He also wants to know, is it fair to say that the history of bisantrene and of race has recently become even more complex to explain than it already was?
I wouldn't say it's more complex. What it is, there's more of it as we've learned more about bisantrene. So if you think back 6 years ago, we knew nothing about it. We just assumed it was a chemotherapeutic drug, just like all the other, very boring, not very interesting. And it's turned out to be really interesting. So it's become, there's more information, but it actually has simplified things down because we now have one mechanism, one cause that explains what was previously a whole series of just seemingly random things that bisantrene did that we couldn't actually explain. Couldn't explain how is it able to protect the heart and kill cancer at the same time? How is it able to work so well in AML? How is it able to work in combination with tyrosine kinase inhibitors so well, which we've seen in multiple different cancers? All of those things we had no explanation for, and now we do. So in some ways, there's more to know, but the other way, there's actually less that you need to know. You only need to know that it's a G4 binder, and as a consequence of that and has a really strong effect on MYC. That's all the thing you need to take away. Both of those are incredibly important.
Thank you. A couple of questions here on the lung cancer study. So first one is, if you're just about to start clinical development in lung cancer, won't it take around 10 years to get that product to market?
The answer to that is no. That's one of the things that we actually targeted for was accelerated approval. So the last drug that was approved for EGFR non-small cell lung cancer, got approved out of phase 1b, 80 patients. The drug before that got approved out of phase 2 out of about 180 patients. The pressing need is so great in this indication for when the TKIs fail that the regulators are just accelerating these things straight through. So that really means, we don't have that. That's one of the things that we actually really looked for when we were choosing an area to go after, that we didn't have to do 2 large phase 3 trials that are going to take 10 years. We wanted things where we have a big effect. That effect is immediately recognised by the regulators. The regulators say we've got to get this onto the market. We've got patients dying now to go forward with. And this has all happened in 2025, so this is not from the history. So that was part of the selection process of choosing that indication.
Rodney, you look like you had something to add.
Oh, no, really just to note that there are different pathways at FDA. There's fast tracking, breakthrough designation, and all of those just indicate, as Daniel was saying, that the agency there historically has been very keen to accelerate, agents which have significant activity onto the market quickly and get them to the hands of doctors and, into patients to have an effect. So, and you see the same sort of thing in Europe as well. EMA have pathways as well to accelerate, good-looking drugs that are having a clear effect. So, yeah, definitely we're well aware of all those mechanisms, and we will be looking to tap into them as much as we can to accelerate the pathway for bisantrene.
Yeah.
Right. And this talks back to that almost checklist that you had of thinking through, okay, what would be an ideal next indication for RCT?
And I think we shouldn't forget the AML. We're talking about taking that through. That's in phase 3, taking that through to approval. So it's not like, we're 10 years away from a drug being approved. We're taking it through in the indication that it was originally approved for in 1988. This is, I wouldn't say low risk, but it's about as low risk as you ever get in the pharmaceutical industry. To get a drug reapproved for the indication it was originally approved for is, you don't often get that opportunity. I'm sure many pharmaceutical companies would love to have that opportunity.
Right. So a couple of questions here around just interested in what the timeline might look like to get the phase 3 AML approval, Daniel.
Yeah, so obviously it's dependent upon having money. So we can't actually begin that program. So I can't really give anyone a clear guideline on that. Overall, to get through those, to recruit that many patients, you're looking at a few years across that to get to the end of the phase 3. The first stage is really around a year to get through. That's the first 40 patients. So you need to do that, then take all that data, go to the FDA, get agreement on the expansion, and then take that one forward. So it really comes down to a combination. The timeline really comes, you can either go more cost effectively with fewer sites and you progress more slowly, or you run at a very rapid pace by putting on lots and lots of sites, and that increases the cost sort of exponentially. And the balance between those is the difficult part. And a lot of that will be what's actually happening in other indications as well. If the lung cancer program's going absolutely amazingly, then we may want to put more resources into that. If the cardioprotection anti-cancer is going really, really well, we may want to put more resources into that. So those decisions are decisions that we made down the track by the board at the appropriate time, and not today.
Okay, thank you. I'll jump back to a question on the lung cancer study. So one question I would like to ask regarding that planned clinical trial, considering that the largest number of potential patients are located in Asia, it seems a bit odd that only 5 Australian sites in Sydney, Melbourne, and Brisbane are engaged and recruited. Is our Hong Kong and South Korean sites for RC220, are they also planned to get involved in this trial? And if not, why not? Rodney, maybe?
Yeah, happy to address that. I think it's very well acknowledged globally, I think, that Australia is a great place to be doing small, early stage phase 1 studies. We have accelerated regulatory approval, essentially just a notification to the TGA to say we're doing a study. We don't have to go through regulatory review. The setup in Australia is just, fantastic, great doctors, great clinical sites, good quality data. And in that, we're quite advanced. We've been talking to a number of investigators around the country already. They've told us that they're A, excited, B, can recruit, some of them claiming to be able to recruit all at their one site. So, we don't see an issue with being able to recruit for this small study, but absolutely, beyond that, when we're looking into the phase 2, phase 3 type studies down the track, we would absolutely be looking at, broadening the study. We would have to really reaching out to China, Asia, North America, Europe, those sorts of regions as well. And again, we've already identified a number of very good investigators active in the lung space already. So yeah, it just happens that one of them is already on the cardio protection study as well, which is great. So we already have that relationship. So starting something up would be quite easy, I think, in that space. But yeah, from an R&D tax credit, effective use of capital point of view, speed, quality of data, it's really hard to go past Australia. That's the place to start.
And I know if Pete was here, I hope he doesn't mind me speaking on his behalf, he would probably say that he's rarely worked on a program where clinicians have been so excited so early as they have been on the lung cancer study. So I hope that's okay with you, Pete, me sliding that in there.
No, I would add to that,
Yeah.
every single clinician we've spoken to about this has just been wanting to be involved with the program, and because they can see the potential, they know the pressing need, they've got patients, they're responding really well, to these TKIs, and then they start to fail and they have very little to offer them. And lung cancer is one of those diseases that moves really quickly. Once you have no treatment, within a couple of months, it's, very grim. And so, to have not much to offer your patients, people are really looking around. And, the whole idea of using ctDNA, it's really at the cutting edge and that's really attracted a lot of interest as well, being able to optimally use ctDNA to identify the patients that are going to respond best. It has really, one problem I'm not worried about is getting people interested in this program. So at least clinicians. So to date, it's been, we've been scaling our intent down to our needs rather than going out there and having to try and scrape around to get people interested.
Yeah.
Excellent. Okay. Good question here. So EGFR is only one of several TKI targets. Do you see opportunities in other cancers against other targets?
Yes, that's in fact one of the checklists that we had. We wanted to have something that would be a template for expansion. So we can take the same approach that we've used, the same problem exists with all TKI. They work really well for the patients that have the right mutations after a period of time, which varies from TKI to TKI, all the patients develop resistance to the TKI. They have exactly the same problem. And bypass signaling is a major indication that's across, which can be targeted via MYC. So TOP2A may not be very important for some other cancer types, but certainly bypass signaling is really important for all these different paths. And this is something that we have seen ourselves historically. Many of our shareholders will remember we've reported preclinical data in a whole range of different cancers using TKIs, AML, melanoma, kidney cancer, breast cancer, ovarian cancer, and they all worked, and we had no idea why. It seemed to be no which TKI we threw at it. It worked really well, and the reason why is they all sit downstream of MYC. So all these different TKIs that sit on pathways that pass through MYC. So by being able to target MYC, you can target all these upstream pathways. So the opportunity of scaling this out to other indications is enormous. And that's something that's very attractive for any potential partner. If you think about it, pharma wants big opportunities and they want to be able to say, okay, if we spend X amount of dollars on this, say like Merck did with Keytruda, they started from a small indication and they just kept throwing trial after trial after trial after trial at it, finding areas where it would work. And, Keytruda then became, $25 billion a year sales drug. And that wasn't done through selling for melanoma. That was done through expanding it into new indications. So using the same basic approach, indication after indication. And we can do the same sorts of things here with TKIs and other drugs, of course, as well. So I think TKIs is a great opportunity, but let's not forget that there are a lot of other drugs out there that have similar issues.
Yeah, exciting. Okay. What happens if the options aren't converted, Daniel, and how will you fund the programs?
Well, it's not that we won't have to fund the program. We won't start the programs until we have the money. So there's no risk really. The risk is we just continue on with the cardio protection anti-cancer, which is we're fully funded to. The company runs along as per normal, and unfortunately, the shareholders will, not get the benefit of these new programs. I'll be very sad. I think Rodney will be very sad as well. I think everyone at Race will
Yes,
be very sad.
devastated.
But company will continue on and will continue working. So it's not like this is we have to have this, or if we don't have that, we're going to be out of business. No, we're very sensible the way we go about. We don't start a clinical program, unless we've got the funds to complete it. We have the funds to complete the cardio protection anti-cancer program. What we don't have is now the funds to complete these two new programs. But if the shareholders want us to get on with this, they know what they need to do. So
There's
it's in their hands.
a related question here. Why not just do a conventional capital raise to get the trials underway?
As we said, we're always considering all options at all times. A partnership would be another way of funding these things. Partnerships don't come with cost. Remember, you're giving up some of the future value. So the optimal time to do a partnership is not necessarily when you can first do it. It might be you might want to be a bit further along so you don't give up so much of the upside. So there's lots and lots of opportunities and options, and the board will consider all opportunities that are out there to choose the best opportunity going forward. So there's lots and lots of options. This is, I guess, one of the things that makes being a board member hard is you've got to balance all these different things and choose what is the best outcome for all shareholders. All I can say is personally, I'm very heavily aligned with all the shareholders. I want to see a return from my investment, and I want to see all the other shareholders get a return from their investment as well.
Yes, we all do. Okay, so question here on a commercial question. How do you prevent open label sales of RC220 from lung cancer to cardio protection?
You don't. It doesn't matter. Like you charge the same amount for both. So it wouldn't make any difference. So if that happens, off-label use of the drug in a different indication is fantastic. You collect the money no matter how the drug's used.
Okay, thank you. Question for Rodney. With EE bisantrene having the exciting DNA, RNA, G quadruplex binder properties and all the focus on moving ahead with today's discussed clinical programs, is there still a continued focus on advancing the 39 unique FTO protein binding molecules which were announced previously? And are these still as relevant where the focus originally with Monash was on FTO inhibition? Thanks.
And start off in that, I probably will defer to Daniel as well in terms of more strategic aspects of it. But look, I guess we announced the results of that campaign, which I think was successful in identifying a number of potential leads. They were very early stage leads, though, just to be clear on that. They would take some investment in order to bring them through quite a few years of work in order to end up with an actual drug that we could advance into preclinical, non-clinical studies, manufacturing and clinical. So there's still a fair bit of work to do. We need, focused investment in that space. I guess in terms of whether FTO is an appropriate target. I'm not so sure on that necessarily, but I can leave that to Daniel to talk about perhaps at this point. But yeah, they're very early, basically.
Yeah,
Daniel, do you want to jump?
Ultimately comes down to resource limiting. If I had unlimited resources, then yes. If you have limited resources, you have to put it towards the most, the opportunities that are going to generate the greatest return for shareholders. And so some things go on hold until things change. So I can't really say. The answer to that is everything still remains. Whether FTO is a viable target or not, we don't really know anymore. I still think it's got a lot of potential, but we have a drug that basically works like an FTO inhibitor. Plus, it does a whole bunch of other things as well. So it's better for the types of indications we're going after. We want to be able to target multiple pathways so we can avoid resistance. So just narrowing in on one target may not necessarily be the best opportunity. On the other hand, certainly for some metabolic indications, I think FTO has got a lot of potential where you couldn't use something like bisantrene. So, but metabolic diseases at the moment are undergoing an enormous turmoil with the GLP-1 agonists coming through. So it's well, I guess we're kind of blessed with having so many opportunities that you could spin a whole company out of the things that we can't take forward. So yeah, it's a shame. It's just there's just more that we could do than we've got resources to do right now.
Thank you. Okay, I'm conscious we're at 20 past 2, so I think we'll take 2 more questions and then we'll let people move on with their days. So this one, congratulations, Team Race. Can you shed any light on timing of the composition of matter patent fast track examination?
The only thing I can say is what the Australian Patent Office says, which is they give a range of times, and as soon as we have any information, we will update our shareholders. So at this point, but all the advice we've received is that the claims are very likely to be patentable or allowable as the term is provided, but until we get through, we won't know. My personal opinion is it's likely to be a slow process. They'll allow, out of the hundreds of claims that exist, they'll allow some, they'll block others, we'll argue with them about those, they'll bring up irrelevant papers and patents, and we'll say, no, that's not the case. Every patent I've ever been involved in has gone through this dance, where at the end of the thing, what you end up getting granted is often very different to what the first opinion of the first examiner is. So ultimately, we have a thicket of different patents and different claims. Any one single claim failing doesn't invalidate the patent position we have, which is a nice position to be in. We've got claims over formulation, we've got claims over the molecule, we've got claims over protection from light for use, manufacturing at scale. All of those go together to really make a very difficult to get around patent thicket.
Thank you. Okay, one last question for you, Daniel, then one for Rodney, and then closing comments. So may I posit the question as to whether the US National Institutes of Health cuts to research grant funding recently create any threats or opportunities to Race? Daniel?
Yeah, well, it's terrible to see, but it probably creates an opportunity for Race. Certainly, our anecdotal experience of talking to US investigators is that not having that money means they're looking around for alternatives. If they can't run a trial being sponsored by the NIH, well then those patients and those clinicians are available to enter into a different trial. So for a company that's coming in with external funding that's not dependent on the NIH, it should make it easier to bring things forward because you have less competition for patients and clinicians. But ultimately, if it destroys the infrastructure, then it may make it very difficult to run any clinical trials at all. So yeah, who knows?
Okay, thank you. Rodney, does ctDNA act only as a downstream indicator of MYC inhibition, or can it also behave as a biomarker for showing which cases will potentially respond to treatment with bisantrene better?
Well, I guess the ctDNA, it's the shedding of DNA from the cancer cells. So in terms of biomarker, it's a measure of how that tumor is responding to treatment and patient progression and so forth. So what we were planning to do is we're going to focus on the known specific mutation that patient carries. So they get diagnosed when they come into the clinic originally at first presentation. The lung panel is run, and basically they know exactly what EGFR mutation the patient has. So we're going to zoom in on that and monitor that as part of the study, and hopefully it will be prognostic and show activity of bisantrene, of course. We'll also do a more broader panel, I think. So look to see what other mutations might be underlying there at first entry into the study. and also downstream. So we will get an idea, I think, in terms of how mutations evolve whilst patients are on treatment as well. But definitely we'll be using it more as a evaluation of patient response. I don't know if that answers all the question, but yeah.
Yeah, it was a good question too.
Yes,
okay, excellent.
so.
Well, I am moving to wrap us up now. There are a few people that we will get back to that needed follow-up to their questions. But Daniel, over to you for any closing comments.
Yeah, I'd just like to really thank Rodney for the effort that he's put in, to get this program, the EGFR program, in such a short time period up and running. I know he hasn't been alone. He's had enormous support from the clinical and preclinical teams at Race, but I think it's hard to underestimate how much work Rodney's put in and how much effort he has to get it to this point. I gave him the timeline. I think he thought it was a bit unrealistic at the beginning, but he's managed to achieve, everything I hoped he would, and so it's been very pleasing. So yeah, just on a personal note, thank you, Rodney, for all the work. And shareholders, you have no idea how lucky you guys are to have such a great, fantastic team at Race. So yeah. It's a rare team. I think it's been brought together with a really rare drug, and I think we're going to really do something amazing.
Excellent. Well, that's a lovely sentiment to wrap up with. So thank you both for your time. My thanks to everyone who joined. And with that, I'll invite you all to disconnect. Have a great day. Thank you.
Thank you,
everyone.